I'd like to thank the National Academy of Science for organizing this symposium on COVID-19 and for the opportunity to present about the SARS coronavirus 2 and emerging coronavirus as global threats to both humans and animals. So coronavirus belong to the Coronaviridae family with 4 genera, alpha, beta, gamma, delta. They're positive sense envelope, single-stranded RNA virus, the largest RNA virus. And the spike, shown here on the surface and schematically on a particle, encodes the virulence receptor binding domain epitopes and is a vaccine target for virus-neutralizing antibodies. Coronavirus infect mammals and birds. They cause mainly respiratory and enteric disease, but other syndromes depending on the coronavirus strain and species. Interestingly, some of these other syndromes resemble the sequelae reported after COVID-19 infection of humans. So all coronaviruses have a similar genomic organization, and they encode 4 major structural proteins, but they encode a variable number of these open reading frames shown here in purple, and these encode accessory proteins. Many of these are involved in inhibiting the host innate immune response, and for others the function is unknown. So coronavirus, it's thought that birds and bats are natural reservoirs for coronavirus, that interspecies transmission can spill over from either bird or bat reservoirs, and also the reservoir host, there can be recombinants occurring within different species within these reservoirs. So we have the Coronaviridae family depicted here, alpha coronavirus, beta, gamma, and delta. I'm going to focus on the ones, the zoonosis ones that have spilled over into humans, but we have a large repertoire of species for both alpha and beta coronavirus, as well as delta and gamma. So we have first the 229E coronavirus, the endemic common cold coronavirus spilled over. It's estimated it spilled over in the 17 to 1800s from bats through alpacas into humans, and predating that was the NL63 from bats to humans, estimated in the 12 to 1500s. And then we have beta coronavirus that have spilled over from animals to humans. The OC43, it's estimated this virus spilled over in 1898, all based on molecular clock analysis. And there was a large pandemic reported during that time called the Russian flu, and it's possible that this could have actually been the first coronavirus pandemic. So the endemic common cold coronavirus are 229E and NL63 alpha coronavirus, and then the beta coronavirus, OC43 and HKU1. So then going on to the Sarbeco and Merbeco coronaviruses, we have the SARS coronavirus, and then the related SARS coronavirus 2, and then we have the Merbeco coronavirus, the MERS coronavirus, and the chronology here is the SARS coronavirus spilled over from bats through civet cats, raccoon dogs in the marketplace in China in 2003. And then the MERS coronavirus appeared about a decade later in the Middle East. This thought spilled over from bats to camels to humans, and this is still ongoing in the Middle East. And then in 2019, we had the spillover potentially from a bat reservoir of the SARS coronavirus 2. We don't know about the intermediate host. We're also seeing spillover events into livestock, and I'm going to give examples of these recent spillovers in swine. So the questions I'd like to address are, why are coronaviruses so diverse, and how did they emerge and evolve to infect new hosts? And then which coronavirus crossed the species barrier? And then what are the animal reservoirs for COVID-19? How are they transmitted from animals to humans and vice versa? So the first question about the diversity, the emergence, and evolution. The coronavirus have a large RNA genome, so this has high plasticity to accommodate changes. There are recurrent deletions throughout the genome, but especially in certain regions, such as the S1 N-terminal domain. This can alter antibody epitopes, co-receptors, but this cannot be corrected by the proofreading activity, which the polymerase of coronavirus possess. The coronavirus occurs as a quasi-species or a swarm of viruses in a population, and this could explain the origin of interspecies variants with altered tissue or cell tropism. I'm going to talk about the pair, the enteric and the variant of the enteric strain in swine, the porcine respiratory coronavirus. We had the feline coronavirus with an altered tissue tropism from enteric epithelial cells to macrophages, which gives us a systemic and provides a systemic infection, and this then results in feline infectious peritonitis in some cats that's fatal. We also have high recombination frequencies among coronaviruses. So it's thought that these mutations, deletions, and also recombination events have led to the emergence of new strains, including new zoonotic strains. So I want to give an example of the interspecies zoonotic transmission, first for alpha coronaviruses. So these are the alpha coronavirus 1 species. There's a reservoir community of these genetic host range mutants in cats, dogs, the swine, TG lineage, and then I'm going to mention a human case that was just discovered. All of these viruses share the aminopeptidase N receptors, so co-infections are possible. And recombinations, mutations, deletions have occurred with interspecies transmission, giving rise to new strains. So an example is the feline coronavirus 1 in a double recombination event with the canine coronavirus. This led to a new strain of feline coronavirus, the feline coronavirus 2, and this changed the pathogenicity and also the antigenicity. We had the origin of TG from the canine coronavirus 2, and then after this there was a recombination event between the TG virus in swine and the original canine coronavirus 2A parent virus, and this led to a new strain in dogs, the canine coronavirus 2, with the N-terminal domain from TGE. And then just recently, we've discovered this occasional spillover of this virus into humans with canine feline coronavirus. And this was first described in a case of human pneumonia from Malaysia in 2018 by Dr. Greg Gray from Duke. And then the virus was characterized further by Dr. Vaslova at OSU. It has a canine coronavirus 2B background. It has a mosaic of the spike, from the pigs, the original recombinant from pigs that's in canine, and then from dogs and from cats. It also has a short mutation in the N protein and a large deletion in the open reading frame 7B that's likely non-functional. Interestingly, the 7B open reading frame was deleted when the virus was transmitted from dogs to swine. So I'm going to also now show you an example of interspecies variants with altered tissue tropism, and a very good example is the change in the enteric coronavirus, transmissible gastroenteritis, to a respiratory tropism, the porcine respiratory coronavirus variant. So TG infects the intestine, it infects virtually 100% of the villus enterocytes, and it can cause fatal diarrhea in baby pigs. On the other hand, this variant, the porcine respiratory coronavirus, infects the upper respiratory tract and the lung. It's a loss of enteric tropism and the high virulence, but it causes an atypical pneumonia similar to that seen with SARS coronavirus 2. And it's thought that the reason for this loss of enteric tropism was related to this large S1 deletion in the N-terminal domain, and this led to the loss of sialic binding, because this is the region where the sialic acid binds to TGE, and this allowed the TG virus bind to gut mucins, but then this was lost in the porcine respiratory coronavirus variant. So we also wanted to investigate if beta coronavirus can cross the species barrier. So Dr. Tsunemitsu, a postdoc in my lab from Japan, we isolated these related coronaviruses from captive wild ruminants, sambar deer, white-tailed deer, a water buck. And then we obtained a diarrhea case from a child with diarrhea from Dr. Zheng, and this virus was genetically very similar to bovine coronavirus. We inoculated all of these into germ-free calves, which do not have a pre-existing microbiota or other contaminating coronavirus, also do not have coronavirus antibodies. All of the calves developed diarrhea. All of them shed the virus in the feces, and all of them seroconverted with neutralizing antibodies to bovine coronavirus. So this was one of the earliest examples that these coronaviruses could spill over from wildlife into domestic livestock to cattle, and then reverse zoonosis from human to cattle. And so this is a ruminant coronavirus reservoir community, and when we sequenced all of these viruses, they have greater than 98% nucleotide genome identity to bovine coronavirus. Another worrisome trend is that it's been shown that white-tailed deer, which are part of this reservoir community, are susceptible to SARS coronavirus 2. So WHO has postulated the next pandemic could be due to disease X, and they also indicate this is not a matter of if but when. And it's also possible we will see future coronavirus transmissions to humans. So if we look at some of the red flags for this possibility, the first one are the SARS-related beta coronavirus in bats. There have been 3 major epidemic events with SARS, MERS, and SARS-2, and these latter are still ongoing. It's also been shown that some SARS-related bat strains combine to human ACE2, so there could be the possibility for direct transmission from bats to humans. The next virus, the porcine delta coronaviruses, were discovered in pigs in Hong Kong in 2012. However, they caused a major epidemic in US swine in 2014 and caused vomiting, diarrhea, and death in baby pigs. So with collaborators at Ohio State, we showed that these viruses can have broad host range. Dr. Kinney's group showed they can infect poultry, including turkeys, and cause mild disease. We showed there's some infection in our germ-free calf model, and also, of course, swine. So in collaborative studies with Dr. Bosch and his lab in the Netherlands, we found that these viruses all have the APN receptor, just like TGE, and they can infect human cell lines. And then a recent preprint study suggested infection by porcine delta coronavirus in Haitian children with mild febrile illness. And interestingly, one lineage was from the Chinese lineage and another was from the US lineage. So this is concerning because this could be the influenza type scenario where you have reservoirs for delta coronavirus in birds, and this time it's songbirds. You could have spillover then to the intermediate host with the porcine delta coronavirus, and then potential for spillover to humans or other species. So the third virus is the swine alpha coronavirus, severe acute diarrhea syndrome epidemic in pigs in southern China. There's genetic evidence for direct transmission from bats to pigs that killed about 25,000 baby pigs in southern China. And more concerning is that this virus was shown in Dr. Baric's lab to infect primary human lung and intestinal cells. So going on to possibility of COVID-19 zoonosis, some of the early cases had contact with live animals in the animal markets in Wuhan, China. And so what is the most likely host animal reservoir for SARS coronavirus 2? It's, again, these rhinolopus horseshoe bats. And at least 3 bat coronaviruses have now been shown to be the most closely related to the human SARS-2. And this one RM strain also has a purine cleavage site in the S1, S2 that was not present in SARS, but is present in SARS coronavirus 2. And now there's also reports of close related bat strains from Southeast Asia, from Thailand. So then another question, is there an intermediate animal host that transmitted the virus to humans? Some suspect the pangolin because it has a similar receptor binding domain, similar high nucleotide identity to SARS-2. But overall, the genome identity is much lower, 91%. So other possibilities are small carnivores or muscelids in the wild animal markets. Others have shown that these raccoon dogs are also susceptible to experimental infection with SARS-2, just like they were with SARS-1. So another question is the possibility of mink and pets as secondary hosts. For mink, there have been major outbreaks in farm mink in Europe as well as the USA. There's rapid transmission among mink and millions have been culled. But importantly, these mink also showed similar signs to COVID-19 in humans, including interstitial pneumonia in some and death in some. More worrisome is the fact that there have been cases of spillover into wild mink, one case in Utah that was close by a mink farm, but two cases, possible cases in Spain that were distant from any mink farms. So initially we had reverse zoonosis spillover from humans into mink, and then we had spread in the mink and zoonotic event spillover back into humans. So in Denmark, there have been 214 human SARS coronavirus infections with mink-related variants. And these cluster 5 variants are of the biggest concern because they have 4 genetic changes in the spike and at least one in the receptor binding domain. And the concern is these could affect transmissibility, diagnostics, or human vaccines. So the question is, will vaccines be needed in the secondary host to prevent new variants and spillovers or reservoir community? And this might also be the case to control the MERS coronavirus by vaccinating camels. Going on to pets, the secondary host in cats and dogs, we have the pre-variant emergence in terms of the coronavirus circulating in humans. There is no evidence of transmission from pets to humans, but there are cases where pets and dogs, cats and dogs, were infected in a household of COVID-19 cases. So to investigate this, there were experimental studies done in cats and dogs, and in 3 studies, all showed the cats could be experimentally infected with virus transmission to contacts. Some had respiratory lesions and all seroconverted, whereas dogs were only marginally susceptible, but they seroconverted. So then studies have just been published or are in the literature now for post-variant emergence to ask the question, do variants transmit better in animals? So the one variant, that the B117, that is this N501 mutation here in the receptor binding domain, that's prevalent in the UK, was detected in cats and dogs from infected homes in the UK, Italy, and recently the US. And interestingly, these are all preprint articles, but interestingly, pets had increased incidence of myocarditis in the UK study. So another interesting scenario seems to be occurring in terms of infection of laboratory mice. So neither the B1 derivative or the B117 variant were reported to infect laboratory mice, but recently a study suggests that these variants of concern, the P1 and the B1351, the one from Brazil and one South Africa, described up here for the receptor binding domain, were shown to infect mice after the first passage and replicate in the lungs, and also associated with variable disease in the lab mice. So this slide summarizes the COVID-19 experimental and natural infection in animals. All those in green are either naturally infected or experimentally susceptible to infection with SARS coronavirus 2. Those species in white are moderately or mildly susceptible. Those in pink are not susceptible, but now the exception is in the lab mouse studies with the variants of concern. So since these all appear to be spilling over by reverse zoonosis, the concern is we need to have a One Health approach with multidisciplinary teams to look collectively at human health, animal health, and also environmental health in terms of these spillover events. So the question here is, are the SARS coronavirus 2 variants more transmissible to animals? So finally, to conclude, in terms of coronavirus interspecies transmission, we have bird reservoirs for Delta and Gamma coronaviruses. In the Midwest, we have these large open feed bunks with corn, and we often see congregating birds here. These are Canadian geese in large flocks. These are starlings. And this corn then is fed to livestock, including swine, and the concern there whether this could lead to a spillover event of the porcine Delta coronavirus from birds to swine. We also have bat reservoirs for the alpha, beta, B, and C lineage coronavirus, the Imbeco, Sarbeco, Mirbeco subgenera, and then ruminants and rodents for the Imbeco bovine-like clade A coronavirus. So why birds and bats? They're multiple species. They're high density, as you see here in these photos, and they usually have subclinical infections, so they're migratory. They can spread the virus long distance. So concern is the coronavirus adaptation to the new host with interspecies transmission, and may result in expansion in the new species, and then there may be appearance of interspecies variants like we're seeing now with COVID-19. There may end up being multi-species transmission to other animals, which then could establish a new reservoir host community for COVID-19. And what this might look like is in the center picture here, a beta coronavirus COVID-19 host reservoir. If this spills over into many different animals, including pets, wildlife, or even livestock.
It's a pleasure to be able to speak to you today about coronaviruses. I'd like to note my conflicts of interest at the bottom of the slide. And today's talk is going to focus briefly on coronavirus genome organization and its transcription strategy, segue into coronavirus RNA recombination and coronavirus host susceptibility loci. The goal is to blend these into an understanding of coronavirus evolution and pathogenesis with some focus on the new SARS coronavirus 2 variants of concern. And obviously, this is a coronavirus particle and a schematic diagram of the coronavirus, which today's talk will focus on. Now, coronavirus transcription is among the most complex of any of the positive strand RNA viruses, and the genome of the virus is about 30,000 nucleotides in length, roughly divided into two domains. The first 20,000 nucleotides of the genome encodes the ORF1 and ORF1b proteins that encode the RNA-dependent RNA replicase and other replicase proteins. These replicase proteins drive the synthesis of a series of subgenomic messenger RNA that are arranged in the form of a nested set from the 3' end of the genome and that extend to various distances toward the 5' end of the genome. This organization allows for different downstream ORFs, like spike, 3A, 3B, and E and M, and so on and so forth, to be placed at the 5' ends of different messenger RNA, so they'll be in the position where they can be readily translated into the viral proteins, including the structural proteins, S, E, M, and N, as well as luxury ORFs that play important roles in viral pathogenesis. Now, interestingly, coronaviruses use a discontinuous transcription program to make these mRNAs. And in fact, at the very 5' end of each messenger RNA is a leader RNA sequence of about 70 nucleotides in length, which is derived from the leader RNA sequence at the 5' end of the genome. And this leader sequence is adjoined to each of the messenger RNAs at these body transcription regulatory sequences, or TRS sequences, the red box element shown here, to drive the expression of each of these mRNAs. And there's a conserved sequence, ACG, AAC, that's present at these junctions. Now, the coronavirus transcription strategy is complex. Polymerases bind to the 3' end of the positive strand and begin to synthesize subgenomic negative or negative stranded RNAs. Some of these negative stranded RNAs will pause at the first TRS element, while others will go to the next TRS element and pause, while some will transcribe through all the way to the 5' end of the genome. These incomplete negative strands terminate in a complementary sequence, which can hydrogen bond with the TRS sequence present at the 3' end of the leader RNA sequence, allowing these RNAs to prime the synthesis of an anti-leader RNA to be present at the 3' end of their RNA molecule. This mechanism spans a jumping event of about 20 to 25,000 nucleotides in length. Some think these incomplete RNAs jump the distance. Most of us believe that these distances are actually quite small, and the body TRS are held in close proximity to the leader TRS by high-ordered structures and protein components that loop out these intervening sequences and allow for slippage across these gaps. Another novel hypothesis came up that was proposed by Luis and Juanes's group is that the 5' and 3' ends are coterminal and held together by viral proteins or other proteins. When the viral polymerase binds to this protein RNA complex, it begins transcribing, but it holds the 5' end as part of that template as it begins to move down the strand. As a negative strand template is synthesized, a choice occurs at each TRS element where it can continue to read through that element or switch templates and read the leader sequences from the 5' end of the genome. Now, how is this programmed recombination event regulated to drive mRNA synthesis? It's really not known. How the TRS sequences are brought in close proximity is really not known to program this strand switching event. In any event, following the interaction of the incomplete RNAs with the leader TRS at the 5' end of the genome, a full-length negative subgenomic negative strand is synthesized, which drives the expression of multiple copies of the subgenomic messenger RNA, leading to the presence of a series of both full-length and subgenomic length messenger RNAs. In addition, at about 1/20th the concentration, there's a series of equal size subgenomic negative stranded RNAs and full-length negative stranded RNAs. But if you deep sequence these elements, you find large numbers of low abundant leader transcripts and anti-leader containing subgenomic RNAs, some of which can encode novel out-of-frame ORFs that are present in the genome. An undiscovered question in coronaviruses is whether these novel ORFs can code novel functions, whether they're protogenes from which evolution can evolve new functions, whether they're irrelevant, or whether minute variation in variants of a concern that emerged during a pandemic can have minor changes within these elements that can enhance the amount of these non-canonical transcripts that would encode more of the novel ORF that could affect pathogenesis. In addition to program transcription by, in essence, a strand switching template mechanism, coronaviruses also undergo RNA recombination at high frequency during mixed infection, and they approach 25% of the progeny of a mixed infection can be recombinants. This not only occurs in a laboratory setting, but it occurs in the natural world, and it just basically is shown here where a polymerase template is moving down the positive strand. It switches to the genome length molecule of a heterologous coronavirus and makes a chimeric negative stranded recombinant RNA molecule, which drives the synthesis of recombinant genomes. Now, importantly and uniquely, coronavirus recombination rates seem to increase from the 5' to the 3' end of the genome. And that's because not only full-length templates can participate in strand switching, but full-length and subgenomic length templates can participate in strand switching. So in this model, a polymerase is making a full-length plus strand of one genome RNA molecule. There's a switching event where it binds to a subgenomic negative stranded RNA and then completes the synthesis of a full-length genomic recombinant molecule. So recombination can occur both during positive and negative strand synthesis, and it can also drive higher rates of recombination at the 3' end of the genome because there's more template available. Now, what are some of the biological constraints that prevent recombinants from being viable? Well, one is the transcription regulatory elements. In this case, we have a coronavirus with a red box TRS elements that are shown here that are regulating subgenomic transcription. And you can use reverse genetics to make a virus that in essence has blue box TRS elements with a different sequence. both viruses are completely viable. Although as you change these blue box elements to more and more unique sequences, they begin to become attenuated. Recombinants between the red box and the blue box element viruses are actually not viable because the leader TRS cannot function with these synthetic TRS elements across the genome. So coronavirus TRS sequences can regulate the capacity to recombine coronavirus genomes across the family. It also can be used as a strategy to build recombination-resistant live attenuated viruses that are safer than normal live attenuated viruses. What else do we know about coronavirus recombination? Within a subgenus like the sarcoronavirus, many recombinants are viable. Structural proteins appear to be interchangeable. Recombination events can occur naturally, virtually at all locations across the genome. When you begin to look at cross-genic group recombinant viruses, it becomes more restricted. We know the accessory ores can be interchanged. We know that some of the structural proteins are viable in certain combinations, but many of these cross-genotype recombinants are lethal, except in the spike gene, where the spike genes between group 1 and group 2 are actually interchangeable. There may be some C-terminal constraints that are involved in assembly and maturation and release, but in general, you can swap out the ectodomain of coronavirus spikes quite easily across the family. So general rules of constraint are shown here. In general, TRS element networks and protease networks are key for viability as well as the functional polymerase. And in essence, more recombinants, as you get more extreme, are more likely to be viable at the 3' end of the genome. Now, another important driver of coronavirus evolution is the mutation rate. Mutation rate is around 10 to the -6. They're 1 out of every million bases is a mistake. This was important for production of for viability of the large coronavirus genome. And they have a regulated fidelity component called NSP14, which removes mismatches from the 3' end of growing nascent strands. One thing that we don't know is whether these fidelity rates are under regulation so that they can be adjustable depending on the environmental scenario, such that when they need to evolve quickly, the fidelity rate is reduced or increased when they need to be stable, when they're in a stable scenario. Another important constraint, a driver of evolution, is just the plasticity of the spike glycoprotein. It tolerates high rates of mutation, deletions, insertions. The structural motifs within the spike glycoprotein are interchangeable between strains, and this affects host range tropism and transmissibility. Now, we've seen the emergence of a large number of new variants since about recognized around May of 2020, starting with the G614 variant in the spike glycoprotein. This variant enhanced transmissibility not only in humans, but also in hamsters. It was 2 times more sensitive to neutralization because the RBD, the part that binds the ACE2 receptor to mediate docking and entry, is in an open position. And so this virus was much more infectious. It was also the progeny, the parent, to all the existing variants that we talk about today, like the UK variant, the South African variant, and the variant in Brazil. These variants also have increased transmissibility. They can escape immune neutralization responses by 2 to 10-fold, depending on who does the assay, and they have enhanced virulence, at least some of them do, in humans, including the UK variant. We see deletions and point mutations across the spike glycoprotein, as you would expect, but what we don't know is what are the limits of these phenotypes. Well, one way to look at this was done by David Martinez in my lab. He vaccinated mice with the SARS-2 mRNA spike vaccine, which gave new titers of around 1 to 10,000. And then he tried to neutralize clade 1 SARS coronavirus, SHC014 and WIV1 viruses, noting a 40-fold reduction in neutralization, 50-fold reduction in neutralization, or 300 to 500-fold reduction in neutralization with these strains that are more distant. And in fact, the SARS-2 mRNA vaccines actually don't protect against SARS coronavirus 2 challenge. So there's enormous potential for increased antigenic evolution in the SARS coronavirus 2 spike, and we're really in the early stages. So beware of deletions, insertion, and point mutations, which will likely become more extreme in the future. Now, what about the evolution of virulence? We know nothing about the evolution of virulence in coronaviruses. We know very little about host susceptibility loci that regulate disease pathogenesis. Our lab's been interested in mapping susceptibility loci using the collaborative cross mice. This is an image of the 8 founder strains in the collaborative cross mice, which are bred in a funnel design to produce recombinant inbred strains, of which there are about 80. And by changing the breeding funnel, you end up with different recombination events across the chromosomes and a mixing of different genomes from each of these 8 founder strains, which are shown here in a color-coded fashion. In one CC strain, you might have this and this kind of genome organization. In a second CC strain, it's very different. So you can infect all 80 of these strains with SARS coronavirus and map phenotypes. You can make CC RICs between them, which is an F1 population between 2 different CC strains, and infect hundreds of those. Or you can find extreme phenotypes that show very different phenotypes following infection with SARS coronavirus, and breed 300 mice and infect them, and then look at phenotypes. And in each case, you will see a range of phenotypes, in this case titer, ranging from 10 to the 3rd to 10 to the 8th. Weight loss, 25% to 100%. No weight loss. And respiratory function or antibodies. This is summarizing the current quantitative trait loci susceptibility map of SARS coronaviruses, which includes primarily SARS coronavirus and a clade 2 bat HKU3 coronavirus. Notice that we have susceptibility loci that regulate resident lung immune cells, lung function, lung titer, histopathology features, weight loss, mortality, antibody response, and circulating immune cells. But I want you to focus on this one multi-trait loci here on chromosome 9. This was determined by setting up an F2 cross between 2 extreme phenotype CC lines. CCO11, for example, is highly resistant to SARS coronavirus. SARS coronavirus 2003 and SARS coronavirus 2 mouse-adapted strains. In contrast, the CCO74 strain not only lose weight, but also have high rates of mortality, both with SARS-1 and SARS-2. So you can cross 300 of these, make 300 F2 mice and begin to map loci. So in this particular case, we have hemorrhage loci. These are the 300 mouse strains on the bottom, different phenotypes that occur in terms of hemorrhage scores in the lung. You get a strong quantitative trait loci on chromosome 9, heavily driven in terms of susceptibility by the CCO74 strain. But not only is hemorrhage a phenotype on this chromosome 9 loci, but also respiratory dysfunction, lymphocyte concentrations, neutrophils, overall mortality, weight loss, and weight loss phenotypes. And in fact, the same set of genes present on chromosome 3 in the mouse are syntenic with genes that are present on chromosome 3 in humans. GWAS studies, which identified the same set of 6 genes that are important in regulating severe respiratory disease following COVID-19 infection. Now, we looked at one of those genes, CCR9, that's common between mice and humans, and asked what the effect of SARS coronavirus 2 mouse-adapted lethal infection is. In the CCR9 knockouts, the animals lose more weight. They have increased mortality as compared to the B6 controls. They also have decreased respiratory function for longer periods of time. They have higher virus titers. They have more lung gross congestion scores as compared to B6 animals, shown here in black. They have increased pathology in the lung. This is hyaline membranes, evidence of acute lung injury or acute respiratory distress syndrome. And if you look at infiltrating immune cells, you see increased numbers of eosinophils. CD11B DCs are inflammatory maximonocytes, CD8 T cells, and CD4 T cells, as well as pro-inflammatory cytokines that are involved in the recruitment of these immune cells. So our data suggests the CCR9, CCL25 signaling protects from lethal SARS coronavirus 2 infection. If you knock it out, you see increased numbers of inflammatory DCs, which are known to contribute to SARS coronavirus pathogenesis. This was work done by Stan Perlman. And it predicts this axis actually regulates pathogenesis of coronaviruses, SARS coronaviruses across species. So with that, I'd like to thank you for your attention and thank all the people in my lab, as well as collaborators like and the UNC School of Medicine, as well as WashU with Mike Diamond. Thank you.
Good morning, good afternoon, or good evening. I'm Lisa Ng from the new research entity in A-STAR called the Infectious Diseases Labs, or ID Labs. Thank you for having me. And I'm very happy to share with you on our various studies that we have been working on since last year to understand infection and immunity of SARS-CoV-2 infection in patients and how this enabled us to improve, strengthen diagnostics, and also generate new data points that will enable improved therapies for COVID-19. So we are 1 year on, and where do we stand now? Currently, there are more than 130 million cases with slightly less than 3 million deaths, with most of the cases happening in the Americas, Europe, and parts of Southeast Asia. Now in Singapore, we were one of the earliest countries that started the battle against this pandemic as early as late January 2020. And we went into a lockdown, or what we call a circuit breaker, from April last year all the way to mid-June. And this was instrumental to help control and prevent the outbreaks in the community. At the moment, most of the cases are imported, with a daily number of less than 20. So in total, we have more than 60,000 cases with about 30 deaths. So as I mentioned, we are 1 year on. What is life in Singapore? As much as we try to live normally, but there are new measures. And in the new norm, this is how it looks like, face masks. But other than that, we try to live as normally as possible. In terms of vaccination, we have already started. And the first Pfizer-BioNTech vaccine arrived in Singapore in December. And following that, we had the first Moderna shipment that arrived in February this year. So at the moment, community vaccination is ongoing, and we hope to complete the entire population in the second half of 2021. Now in Singapore, the study for COVID-19 was predominantly driven by the National Center for Infectious Diseases under this study called PROTECT. Now it is a multi-centered prospective study to detect novel pathogens and characterize emerging infections. Now, interestingly, this protocol was not written specifically for COVID-19, but rather it was written for pathogen X. And the protocol was actually put up a couple of months before we had the first case of SARS-CoV-2 infection. We all know now the typical symptoms of COVID-19, which is the fever, dry cough, shortness of breath. However, it is now clearly and well reported that there are more sophisticated or even more severe cases. So in Singapore, typically the disease severity of patients have been divided into 3 main groups, which is what we call the mild cases, which is patients without any pneumonia, just the fever, dry cough, or sore throat. And we have the more severe ones where patients will suffer from pneumonia, but without hypoxia. And the more severe cases that had pneumonia, and some of them then went on into the ICU. However, we also know that there are many reports and more reports are coming up that there are other complications such as neurological, cardiac injury, even thrombotic complications. And all these are mostly sometimes complicated by comorbidities, specifically patients who already have diabetes or even cardiovascular diseases or hypertension. So there's still so much more to look at in order to understand how this virus actually generate more severe outcomes. Now in Singapore last year, when we had the first outbreaks, we had to work closely together, as I mentioned, for the PROTECT cohort, and this involves multiple institutions and also hospitals. Over at A*STAR, the immunology work was driven a lot previously by the Singapore Immunology Network and now the ID Labs, so with my co-lead investigator, Professor Lohan Rainier. So the idea was really to have a deep understanding of the various aspects of infection, using various approaches such as flow cytometry, mass cytometry, immunogenomics, proteomics, or even immune imaging to understand the various immune phenotyping of this infection. And how this would then allow us to identify new biomarkers that could then improve prognosis. And of course, the next thing was also to understand the various mechanisms of the various inflammatory pathways, I beg your pardon, and how this would then enable the possible intervention either with FDA-approved drugs or the development of therapeutics such as human monoclonal antibodies. Now, this is just a very simple a diagram of how the SARS-CoV-2 looked like. Now, it is a very huge single-stranded positive strand RNA virus. And whenever we talk about diagnostics, typically the focus will be very much on the spike protein, the envelope, membrane, and also the nucleocapsid. Now, we all know that in the area of diagnostics and detection, no one method will be the only method. We need various complementary detection systems that will enable us to detect the virus or even the presence of antibodies in the patients at different times. Now, one of the earlier work that the team embarked on was to understand the antibody profile of the patients, and we followed these patients from the early acute infection through the time when they recover and also post-recovery. And are we able to identify specific epitopes on the various antigens from the patients? So what we had done was generate a linear B-cell epitope library against the spike and the viral antigens, and epitope mapping was done. And we found that using a patient's serum or even plasma, we identified specific hit regions in the spike and also the nucleocapsid. And when we identified the specific region to the individual peptide, we were able to identify 4 of these epitopes. Most of them were in the spike and 1 of them in the nucleocapsid. And when we expanded the screen of these epitopes with the patient's plasma or serum from different time points, we found that 4 out of the 5 epitopes could actually specifically detect the COVID-19 patients with good sensitivity. Now, 1 year on, we have followed some of these patients, and you could see from here, while the levels of the IgG were shown to decrease steadily, the presence of IgG could still be detected. And because of that, we were able to identify another epitope that was not picked up in the earlier infection disease state. So we found another epitope, which is in another part of the spike protein. And I'll just move on to the next part of the work. Was now clearly SARS-CoV-2 is a BSL-3 pathogen, and because of that, it requires a stricter level of handling in most laboratories, and that limited the studies and the experiments at that time. So working with the group led by Ching-Yi Wang and his team, they established a safe pseudotype virus assay that enabled the characterization of the neutralizing capacity of these patients' antibodies. So, but before that, we had to do a comparison of how this pseudotype virus assay was performing against a live virus. So working together with our collaborators from the Defense Science Organization, led by Brendan Hansen and his team, SARS-CoV-2 live virus neutralization was done with a selected group of the patient's antibodies. And when we compared the IC50 values with that of the pseudotype virus, you could see that it's a very close comparison. And that enabled the characterization of the patient's materials in a much more efficient way, handling it in a BSL-2 plus condition. And you could see from here that all COVID-19 patients actually elicited neutralizing antibody responses against the virus. Now, subsequently, in the middle part of last year, there have also been reports of new SARS-CoV-2 variants. So the most famous one, it's the non-synonymous mutation from the aspartic acid, the D residue to the glycine, the G, which was found in the position 614 of the spike protein. So this is known as a D614G variant that was shown to increase the infectivity and transmission rate. So over in Singapore, you could see from here that we were also tracking the variants that were happening. And this was really through the collaboration of Sebastian Mauerstro from the Bioinformatics Institute, whom with the use of the GIS-8 were able to track a lot of all these sequences around the world. And in Singapore, the clade pattern, actually typically about 30% of them were in the G clade. Most of them fall into the others. Now, we were interested to know if there was any difference in terms of antibody neutralizing capacity between the G and the D. And you can see from here that using the pseudotype virus assay, we actually showed that patients typically elicited a similar degree of neutralization against both the D and the G pseudoviruses. So indicating that the D614G mutation actually did not really impact the neutralizing capacity of the elicited antibodies. Now, however, as of early this year, it's also been widely reported that there were new COVID-19 variants. So our work is still ongoing to actually screen the neutralizing capacities of the patient's antibodies that were infected last year against these new UK variants and the South African variants. And we do know that the neutralizing capacity has been reduced, just like some of the other reports. So going back to the point of antibodies against the specific epitopes, there were two. One of them was called the S14P5 and the other the S21P2 that stood out. Why so? It is because of where they were located. Now the S14, as you could see, it's located very near the ACE2 binding region, while the S21 was located very near to the fusion peptide. So therefore, we were very interested to see if these specific epitopes had any role in developing the neutralizing capacity of the antibodies. And before that, also we did a correlation of the ELISA against the epitope-based ELISA against the neutralizing capacity. And you could see from here that there is a positive correlation. So therefore, we then went on to study this mechanistically. So the first thing was we tried depleting away the antibodies that were specific against the S14 and the S21P2. And then you could see from here, from the ELISA, that the depletion was successful. And when we then proceeded with the pseudovirus neutralization, you could see from here that the neutralizing capacity was reduced. So this showed that the antibodies that were targeting these two linear epitopes actually accounted for about 30% of the general anti-S neutralizing response. So there is quite a lot of promise to further understand these two peptides. So we move on to the immunopathogenesis of the infection in the patients. And you could see from here that it's led by a larger team of researchers that went on to study the immune profiles. And you could see from here that the profiles actually look different from patients with mild infection to patients with severe infection. And clearly there is a pro-inflammatory signature, what we call the cytokine storm. And this has just been recently updated, where when we followed patients that were 6 months post-infection, you could see from here that the inflammatory markers typically actually reduce significantly. But yet, we were able to identify or separate the patients into different groups according to their disease severity. And then there's another part of the story, which was the identification of a specific deletion of the virus. This was first reported by the team of Gavin Smith at the Duke NUS, where they found the deletion of the ORF8 in a specific group of patients early on during the outbreak. But of course, with the strong control of the outbreak, then this mutant virus was also controlled. But it's interesting because as we compared the immune profiles, you could see from here that patients with the deletion actually had a milder or rather more muted pro-inflammatory signature. And also this then corresponded to the milder clinical phenotype as well, where patients had milder disease. So of course, we then went on to study the protein-protein interaction network that actually found several host factors that were shown to associate with the ORF8 that suggested various possible mechanisms of how this could control the outcome of the infection. So I'll move on to the last part of our immune phenotyping study in patients, and this is done by the flow cytometry. And by just using a FACS panel, a wide FACS panel of more than 50 markers, combining the myeloid panel, granulocyte panel, and the lymphoid panel, we were able to actually show the differences in the patterns and what are the immune markers that were upregulated during the acute infection. And actually, we showed that there was increased expression of CD169 and also CD11B from the acute severely ill patients. And of course, these markers then went back to almost normal after when the patients recovered. So you could see from here, using the heat map representation, we found that there was a high number of monocyte activation and also neutrophils activation. But upon further investigation, we actually showed that by looking at a specific population of the neutrophils, what we call the immature neutrophils, we were able to see clear differences from the acute patients. So by looking at just this subset. And so we went on to also assess the asymptomatic patients, which then this started second part of last year. And using the same approach, we showed that the immature neutrophils between the asymptomatic and the healthy controls was not quite significant, not that strong when we compared with the symptomatic patients and what you've seen earlier. So we also perform whole blood transcriptomics against the patients. And you could see from here that there is a clear difference between the asymptomatic and symptomatic patients during the early stage of infection. So there's still a lot of work to be done. And I will end off my talk here for today, and happy to take on any questions later on. And I would just like to acknowledge the various members in all the various institutions, from the Ministry of Health, all the different hospitals, Duke NUS, the Defense Science Organization, and also the other colleagues at the A-Star. So thank you so much for your attention.